Okay. So embryology is the part of human anatomy where we are going to learn about development of a human from a single cell stage, even before forming the single cell stage, from where exactly these cells are getting ready and how they are meeting to form a zygote. That is what we are trying to study in embryology. We will be studying general embryology. There are two sections in embryology, that is general and systemic embryology. In general embryology, we'll be studying around till 4 to 5 weeks of development, where from a single cell stage till 4 to 5 weeks, what are the series of events we will be studying. In systemic embryology, we are going to study about every system, how it develops, like limbs, okay, GIT, reproductive system, urinary system. That is how we should be studying. For dental students, we don't teach all systemic embryology. We teach only head, face, neck embryology. Okay. So that's why you'll not be studying about the entire development. We will be restricting your embryology section till general embryology, and the last part is only head, face, neck embryology. This will be just an introductory session where you will be learning about certain terms related to embryology and a little bit in details about cell division. Okay. So what is the meaning of embryology, we will see. And if you have any questions, please stop me so that we can discuss, we can we can talk about it in detail. Okay. Till last class, any questions? Anyone wants to ask something? Okay. So are you all able to hear me? No problem, right?
Yes,
Yes,
ma'am.
ma'am.
Yes,
Yes,
ma'am.
ma'am.
Yes.
Okay,
Yes, ma'am.
fine. Now, what is the meaning of embryology? See all these cute babies. We see these cute babies, okay, and we feel that, yeah, they are so beautiful, very pleasant to see them. But there are a lot of complex events which are going on, and a lot of genetic manipulations which goes inside human system to give rise to this such cute babies. Okay, so we are going to study how these cute babies are born like this and how they are developing. Let us see what are certain terminologies related to embryology. Okay, embryology means study of embryos. However, the term generally refers to prenatal development of embryos and fetuses. Prenatal means before birth. So we are going to study embryology. Usually the right term is developmental anatomy, where we study development of a human being. But since we are trying to study about embryos and fetuses, we usually use this term embryology. Okay, so what is a developmental anatomy? It is same, refer to embryology itself. But if you actually see scientifically, embryology means where we are trying to assess the development of embryos, only embryos. Okay, not concerned with the development of placental tissue, not concerned with the development of internal changes or physiological changes in the human body or the mother's body. We are only concentrating on the embryos, which is scientifically referred to even development in vitro fertilization, where how the embryos are grown outside. So everything is coming under embryology, but developmental anatomy includes everything. Okay, so it includes the maternal changes, it includes the hormonal changes, it includes even the changes in the fetus, from starting stage, from a single cell stage, and even before that, how the human body is developing to get ready to give birth to a new organism. So like that we study everything under developmental anatomy. So you will see all these two terms in sync with each other. Now why we have to study embryology? What is the importance of it? Okay, why not directly just study medicine and get out with it? Okay, why we have to study embryology? If we understand embryology, we understand the basis, okay, how a structure is present in our body. Okay, for example, here we are showing the structure of a stomach. Initially we can see that the stomach is like this, a small swelling, a tube, a tube shows bulging. Okay, now because this is a longitudinal axis of the tube, now what is happening during development, there is a rotation process going on. Okay, as we had discussed before also, upper limb lower limbs they rotate, right? So same like that, the stomach also rotates. Because of the rotation, what is happening, initially the anterior border and the posterior border of the stomach becomes, anterior border becomes right border, okay, and posterior border becomes the left border. Okay, now what is happening, because of this rotation, the anterior border becomes shorter and the posterior border, now it is no more anterior and posterior, it is left and right borders. So left border becomes shorter and the right border, sorry, right border becomes shorter and the left border becomes enlarged, and the stomach grows in the size towards the left side. So that's why we get a J shaped stomach, okay, with two curvatures, lesser curvature and a greater curvature. Now you need not bother about all this, need not remember all this. What is important for you, why we have to study embryology? Why we have to study embryology? Because why we appear in a way we appear. Okay, for example, how our eyes are coming to front of the face. Okay, how upper lip is formed in this way, because initially uh, two nostrils, one is on the, now where are your ears, there usually we will see nostrils just above that eyes. Okay, and later on the nose, the two sides. Okay, so that means two eyes are coming forward, joining with each other. Okay, joining, coming closer to each other, so that what is happening, the distance between the eyes is getting closed. Okay, is not completely closed, it is becoming narrower. Okay, so that's why the eyes are coming to the front of the face. Same like that, upper lip, it is not formed as a single entity, it is formed separately. Okay, one on each side, later on both join together to form a very nice single unit of upper lip. How it is developing, why it is developing, we should know. Why we should know? Because we have certain kind of anomalies. Okay, so we have certain kind of if this is not happening properly, we might have some kind of difficulty in a newborn baby. So what are these difficulties? Are called as embryological abnormalities. Embryological abnormalities are called as anomalies. Okay, see, for example, these are anomalies. Now, a child might come to you with some kind of anomaly, or a child is born with some kind of abnormality. This is called as polydactyly. Okay, I told you, numerous fingers. Okay, rather than usual, we have extra fingers. Polydactyly, sometimes less fingers. Okay, so like this, when we are looking at certain kind of abnormalities, we try to question why this is happening. Okay, when all the children are born, all newborns are born normally, why few children are not born normally, why they are having anomalies? So to understand that, we need to understand what is the embryological basis of development of every structure, then only we'll come to know. For example, as a dentist, you might ask, why is it important for me as a dentist? So for you, you might see some people with hair lip. Hair lip means, probably you are all aware, small cut in the upper lip, right? So palate, sometimes palate is not fused properly. So what happens is, there is a space between the nose and the mouth. Okay, there is a passage opened from the nasal cavity to the oral cavity. Sometimes it may not be completely closed. Okay, you might see some children around with some kind of abnormalities in the head, face, neck as well, because nervous system developmental abnormalities and face abnormalities are linked with each other. So these two are, whenever you see such kind of abnormalities, you get to know that yeah, some kind of embryological um, development has been stopped. Okay, so that's why when we look at some patients, when we need to know, before treating them, we need to know what has gone wrong. Okay, what step, what process have gone wrong, and can it be corrected or not. Okay, so like let us say, when we are talking about development of organs, okay, we need to understand that yes, why a particular structure supplies a region. Okay, for example, stomach, if you see, we have right and left vagus. Always in our human body, we have right vagus nerve and left vagus nerve, two vagus nerve. These are the cranial nerves, okay, one on each side. Now if you see stomach, it's actually on the right vagus and left vagus are present on both sides. Because of the rotation, what is happening, the right vagus goes to the back side and it supplies to the posterior region of the stomach. Okay, whereas left vagus comes to the forward. Okay, because initially it is on the left side, but because of the rotation it comes forward and supplies to the anterior surface of the stomach. Okay, so like this, some kind of changes are happening in the body. Okay, why that positions are changing? Because of the development. Okay, if you see, git rotations are very very common. Okay, what happens is the liver is initially formed in the middle. Okay, because there is a single tube in the abdomen, and that tube is growing long, and from that tube, like a small buds. Okay, how the flowers are growing, from a branch, same like that. From this tube, small small buds start growing. Okay, like liver bud, gallbladder, okay, spleen, pancreas. These are all extra parts which are developing from this single tube. So that's why we need to understand the embryological orientation. Now let's see this. For example, we also understand, if we study embryology, we also understand what are the kind of variations which are happening in human structure, and also birth defects. Okay, so birth defects, like for example, some kind of medication can cause a birth defect. Okay, so if you see, this polydactyly, these anomalies are quite common because of what? Because of certain drug, initially in 1900s. If you see, there were some drugs which are given to reduce the um, vomiting sensation during pregnancy. Okay, so severe nausea and vomiting sensation will be there for, because of the hormonal changes at the initial stages of pregnancy. Now to stop that, they used to give certain drugs, and because of that, many children during that phase were born with this kind of anomalies. So that is when we came to know that yes, the drugs might affect the development of the embryo. So that's why we need to take certain precautions before going for even any kind of medication during pregnancy. Okay, so we will come to know even infectious agents, x-rays, drugs might affect the human development. Says here, this is unilateral amelia. Amelia means no limbs. Okay, miromelia, you can see that very small limbs, abnormal limbs, you will see like this, a child with this. So you can see that irregular bones. Okay, all this, these are the normal anomalies because of the drugs which you take during pregnancy. Okay, so also embryology will provide what? Embryology will provide essential knowledge for creating healthcare strategies. Healthcare strategies means yes, we need to understand now what happened. We need to find out whether the child, whether the baby which is growing inside is normal or not. Okay, because if it is not normal, we can take certain precautions before birth itself. Now if it is normal, then no problem. That's why what happens, we started designing systems to understand how the human which is growing inside is appearing. Okay, like ultrasonography, okay, fetal MRI. So all these technology were invented to find out the fetal health, in vivo fetal health. Okay, so that's also is a part of embryology, where we will understand fetal sonographies, how a fetus appears at every single week, what kind of changes may happen, we can study from outside itself, not just sonography. See, this is in this picture we are trying to do a chorionic villi which are present here. We can take out amniotic fluid. This is called as amniocentesis. Why do we do this amniocentesis? When we know that, yes, for example, mother and father are having some kind of genetic disorders, okay, or mother or father, anyone is having a genetic disorder, like let us say hemophilia. Okay, I hope all of you are aware of hemophilia. It's a blood clotting disorder. So something like that, if a parent is having that and it is possible that they want to know whether the child is having the same gene or not, so then we can go for amniocentesis. What do we do in amniocentesis? Ultrasound guided, this one, the chorionic this sampling process we'll do here, where the needle is injected into the amniotic sac, where amniotic fluid can be removed, as well as we can take out a sample of chorionic tissue and we can try to do a karyotyping. With karyotyping we'll try to understand the genetic composition of the fetus. So during this time when we do genetic composition, that means we plate that all the chromosomes, we can see where any abnormalities are present or not, or on which gene there is an abnormality, and we can predict how the child is going to be present after birth. Okay, so all these processes can be seen before birth itself. This technology has been very much advanced now, so we'll get to know what is the health status of the fetus in vivo. Okay, so any questions still here?
No, ma'am.
Oh,
Ma'am, do we have to remember different types of abnormalities?
no, no, not now. Now you just need to understand what is going on. This is a general introductory lecture, but after, when you are studying, wherever it is required, we will tell you, okay, which topics are important, which will come as a short note question, we will tell you. So those things, yes, you have to study. And when you are talking about abnormalities, usually we head face neck developmental abnormalities we will be teaching you, so those things you will have to remember. Rest of the things you need not. Okay, whatever is important I will stress on that, so need not worry about it. Any question?
Ma'am, you yes, ma'am. You told about the
Yes, someone is questioning.
uh, so
Yes, go ahead.
hello,
Yes,
hello. Yes, ma'am. You told about that.
I can hear you, please go ahead. Okay, um, amniocentesis, one of you has
No.
asked a question. Amniocentesis is banned. Yes, normally it is banned until and unless we know that the child might have some kind of genetic abnormality, or the fetus is having is going to or expected to have some kind of genetic abnormality because of the parents. Okay, or during routine ultrasonography some kind of abnormality is detected, then we go for amniocentesis. Okay, it's not banned, it is banned from routine procedures. Okay, it is still done, it can be done, but in India, because genetic what to say, the karyotyping of the fetal tissues is not allowed because of the sex determination. Before birth itself we can find out it, because we are trying to take out the genes, right? We are trying to take out the chromosomes and trying to find out. So if we can, when we play it, we'll come to know whether the child is going to be a male or female child. So that's why we have certain kind of PCPNDT act. Okay, PCPNDT act. Okay, so this act says that yes, you should not disclose the sex of the child, because sometimes there might be problems, because sometimes what happens is, in India, it can be possible outside, out of India also, it is possible that the babies can get aborted because it's because of the sex determination basically. So that's why PCPNDT act says that even if you're doing chorionic amniocentesis, we will not disclose the sex of the fetus to the parents. Okay, that is a rule. Do we need to buy separate book for embryology is a question someone asked. Yes. Okay, that book I will tell you what you have to buy right now. Don't buy anything. Okay, uh, we have a separate book for embryology. Okay, now someone else asked the question, genetic disorder in which the hands and legs are of abnormal, so how does the child survive? Child survives, legs and hands are not essential. Okay, or it is important for the survival of the child. It is possible that the child can live without the limbs, no problem. Yeah, only certain structures are there which are essential for living. So even if those structures are not there, the child can survive. mm, no, doesn't it affect the red bone marrow production? No, it doesn't, because we have several bones in the body, not just the limb bones. So how much is the body, that much requirement is present. So if the limbs are not there, that much red bone marrow is also not sufficient. So I mean, it's not required. So whatever bones are present, that is more than sufficient, right? So any more questions? So I hope all of you are clear with this. Okay.
Yes, ma'am.
Yes, ma'am.
What comes under development? Okay, it's a continuous process that begins at fertilization, when a fertilized oocyte zygote, a totipotential cell, is transformed into a multicellular human being. Fertilization is a process where egg and the sperm are fusing with each other to form a single cell stage called as zygote. We already discussed that in the previous class. What is a zygote? Now, from zygote, which is a totipotential cell, if you remember that, it is totally potent to give rise to an entire human being. So, from a totipotent cell, it is transformed into a multicellular human being. So, this is the development which we are going to study. And during this journey, what we have to do, what we'll study about, how this single cell undergoes lot of divisions. Okay, so we are going to study about stages of cell division. Now after cells are divided, they has to migrate from one place to different places. The cells migrate. Then we also will study about, after migrating, some cells only remain. After that, few cells die. So that is called as apoptosis. What is apoptosis? It's programmed cell death. After its work is over, the cell dies. Okay, this is called as programmed cell death, apoptosis. Next is differentiation. Once a cell goes there, initially it's a simple single cell. Okay, once the cell goes into a certain region, it undergoes into transformation into a specific type of cell. This is called as differentiation. Then we also will study how the cells grow and rearrange themselves. So all this are the different stages which will be studied under development. Okay, so that means, for example, we will take some example. Let us take example of gonads. Gonads means the testis and ovary. These are the gonads. Where they are developed? They are developed in the thorax region. Okay, just below, right now, where we have our kidneys. Okay, there we have our gonads, testis and ovary initially. Okay, now into that only the outer shell is present, but where are the cells coming from? They are coming from different area altogether. These cells are coming from yolk sac, which we will study what is a yolk sac and all you will study later on. Yolk sac is giving rise to GIT, all esophagus, stomach, intestines. From that wall of yolk sac, these cells are coming. Which cells? Germ cells. Okay, gonads are developed in thorax. Okay, thoracic region or lower thorax region, or we can say upper abdominal region. And the sex cells or the germ cells, they are coming from GIT, yolk sac. And these cells slowly divide, they migrate from the GIT, from the wall of yolk sac, into where? Into the gonads. See how perfectly they are synced. They are developing in some place, they're going and migrating and sitting at some place. Okay, and after going there, lots of cells are there. Now these cells are arranging themselves in a certain order. Okay, now these cells, after certain stage, okay, when the fetus is developing to third month, fourth month, fifth month, ninth month, multiple cells slowly reduce, and only what are required amount. Okay, let's say the child, the fetus is a female fetus. Okay, on an average around 10,000 to 40,000 eggs are only present inside, rest all will die. Okay, see that process, programmed cell death. Now only which are present inside, okay, these undergo differentiation. Now yes, they are now settled inside the gonad. Now these cells fate is fixed. Yes, they are going to convert into egg. Okay, oocyte. So now they undergo transformation. This is called as differentiation. So these are all various steps which are going to happen. It's not just in the gonads, it will be seen everywhere. For example, the cells which are forming the palate, the cells which are forming the eyes, the cells which are forming the tongue, okay, the muscles which are inside the tongue, they are developed somewhere else, but these cells are migrating into the tongue to form the muscles of the tongue. How do we know that these are coming from somewhere else? It is coming actually from the lower part of the brain. Now how do we know that these cells are coming from that region? Because these cells carry the nerve from there. Okay, so we know that yes, these muscles which are developed from the cells coming from the lower region of the brain, they carried a nerve along with it. So they are not from here actually. They are not inside the tongue. They are not developed inside the tongue. Only outer cover of the tongue is present there, but inside the muscles of the tongue, they are developed from lower region of the brain. Okay, so that's how we come to know about cell division, cell migration, differentiation and rearrangement. Okay, so I hope you all understood a little bit about this. Yeah,
Yes,
Yeah,
ma'am.
ma'am.
Yes, ma'am.
now let us see what is cellular differentiation. Okay, When we are looking at cellular differentiation, we know what is cell division. Cell division we are going to study. Now, what is differentiation? In differentiation, the cells are giving rise to certain kind of specificity to them. Okay. Initially, we have oocyte and a sperm. They are fused during fertilization and we get specific type of cells in it. Now these cells, already we discussed this. What are the types of cells? These are totipotent cells. Now I pick this cell, one cell from here, keep it in a petri dish and grow them. It will give rise to an entire human being. Okay, any cell you take, it will give rise to entire human being, totally potent to give rise to entire human being. That's why these cells are called as totipotent stem cells. Okay, this is called as potency of a cell. Which type of potency? Totipotency. Then comes to the next group. Okay, now these cells are again dividing multiple times and giving rise to an so many different types of cells here. Okay, cell division. Now after division, now see here there is a group of cells which are inside. Okay, some cells are organized outside, some cells are inside. These cells are taken out. Okay, these are called as embryonic